Hello, good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Ernest Atto from Business Review Webinars, and I will be your host. It is our pleasure to have BioPharmaSpec with us today, who will be discussing on the webinar entitled Characterizing Protein Impurities. Today's guest speaker is Dr. Richard Easton, a Technical Director for Structural Analysis at BioPharmaSpec. I'd like to welcome you to our webinar platform on 24. This webinar is browser-based, so if you disconnect, click the link you receive via email to rejoin the session. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please interact with them all throughout the session. You can ask questions using the Q&A widget or by using the questions box. We will try and get through as many of the questions as we can once the presentation has concluded. Please use the help widget if you experience any technical difficulties or require any more assistance. So without further ado, please allow me to welcome Richard. Richard, over to you.
Thank you very much, Ernest. Good morning or good afternoon to everyone listening. Obviously, the title of the presentation today is Characterizing Protein Impurities. And what I'm going to discuss is the way that we would undertake the analyses of a variety of different protein impurities encountered during product development. Most of the techniques that we'll be talking around are based around mass spectrometry, but not exclusively so. And we'll be looking at various different examples of impurities in terms of product impurities and also impurities that come in through the process of development of a molecule. So first of all, just to give you a little bit of background about BioPharmaSpec and myself. BioPharmaSpec was founded in 2014 and is an independent CRO. It was put together by Professor Howard Morris and Dr. Andrew Reason. Professor Morris has over 45 years of experience in structural analysis, most heavily in the area of mass spectrometry. For 30 of those years, he was associated with the MScan group of companies, which he founded in the late 1970s. Dr. Reason was managing director of the MScan group of companies himself for 20 years. I also used to work for MScan for 13 years. I was team leader for carbohydrate analysis and subsequently principal scientist before joining BioPharmaSpec last year as their technical director for structural analysis. BioPharmaSpec has labs in the UK, based in Jersey, in the Channel Islands, and in the US, in Philadelphia. We offer a full range of structural and physicochemical characterization services to cover the ICH Q6B guidelines, and I will talk a little bit about those later. We use state-of-the-art mass spectrometric and chromatographic procedures to fulfill these requirements, but we also have an extensive array of other techniques to deal with higher-order structural analyses. The scientists at BioPharmaSpec have a very extensive experience of structural analysis, both within the academic and industrial areas. Indeed, scientists at BioPharmaSpec have been involved in pioneering developments for many years in the area of structural characterization, particularly peptide mapping and disulfide bridge assignment by mass spectrometry. And also Professor Morris himself was involved in the conceptualization of the QTOF mass spectrometer, which is now a cornerstone of instrumentation for structural characterization. The scientists at BioPharmaSpec have worked on a very wide range of molecules, and just a list or a short list of biosimilars is shown here. As well as the characterizations of the molecules themselves, we've also been involved in looking at the impurities associated with these molecules on very many bases. And so we have quite a wide range of experience of analyzing different types of impurities, which may all be associated with particular different types of molecule. But why do we need to look at impurities in the first place? Well, quite obviously, the industry we work in is driven by quality. We aim to produce the purest, most structurally analyzed molecules that we can, with minimal impurities present, so that we don't compromise the quality and the efficacy of the product that we're generating. This could obviously have a very deleterious effect on patients. The effects of impurities can be very wide-ranging. They can actually cause aggregation of the molecule. They can affect the properties of the molecule within the cell systems, and within the blood system. But also they can affect the actual properties and stability of the molecule as well. So really the entire profile of the molecule can be affected by the impurities that are present in it. Now, obviously, the guidelines and the regulatory authorities are very well aware of impurities and what they can do, and therefore they require that we do monitor these and that we keep a close eye on what is present in our molecules and that we manage to minimize impurities as far as possible. Now, I've just shown on this screen here the guidelines as shown from the European Medicines Agency and also the FDA. Both of these authorities quote the ICH Q6B guidelines, which are a set of regulatory guidelines that have been around for a fair few years now. But the ICH Q6B guideline is particularly pertinent to this because it covers the analyses and test procedures associated with biological molecules. A section of this is specifically dedicated to the analysis of impurities. And what I've shown on the screen here is a little bit of the text just to kind of highlight the way that the thinking goes. The document states that impurities can be either process or product related, and that these materials should be characterized to the extent possible, which is quite understandable. If we're going to know our product, we have to know exactly what the product is, but also what the impurities are that are associated with that product. Only then can we understand what we can do to minimize those impurities, or indeed what those impurities will do to the product should they stay present. Process-related impurities are derived from manufacturing processes. They will cover things like cell substrates, culture media, and processing factors. Product-related impurities are those impurities that are associated with the actual molecule itself. So these can be precursor forms of the molecule, they could be certain degradation products of the molecule, they may represent forms of the molecule with a variant glycosylation profile, for example, or various other forms that may have arisen during the manufacture of the molecule itself. Now, those guidelines are fairly general, but also in the rapidly expanding area of biosimilar analysis, guidelines have been written to describe how those should be characterized. And again, impurities are also mentioned in these, as you would naturally expect. The quality guidelines from the FDA are shown here, and this states that the sponsor should characterize, identify and quantify impurities in the product and the reference product to the extent that it's feasible. And once again, it also mentions that the product and process-related impurities are as defined in the ICH Q6B guidelines. Likewise, if we look at the quality document on biosimilars from the EMA, the text again says that the purity and impurity profiles of the biosimilar and the reference molecule should be studied using state-of-the-art methods. Also, there is reference back to the ICH Q6B guidelines in terms of the definitions that are used. So there is always a common thread relating back to this document and to the definitions and the texts that are associated with it when we're looking at the impurities that we find in our samples. If we just go back and look at that document in a little bit more detail, we can break down the process-related and product-related impurities into two broad areas. The lists that you see here are not comprehensive lists, but they just list a lot of the common forms and common impurities that are seen. Impurities, quite understandably, can be product-specific, so you need to know what your product is and what the likely impurities are going to be associated with that to allow for analysis to be tailored in particular ways to the molecule of interest. Nonetheless, in broad terms, a lot of these forms are going to be seen commonly in most, if not all, of the biological molecules under development. We're not going to talk about all, or rather, I'm not going to talk about all of those today. We'll just be dealing with the ones that you can see in blue on the screen, either touching on them or in more detail. So first of all, I'd like to start off with looking at host cell proteins and the idea of quantitation of host cell proteins. Quite understandably, the molecules that we produce are coming out of biological systems. Those biological systems will contain an awful lot of proteins that may well be associated in the environment that the molecule is finding itself, that we're actually trying to produce. And these host cell proteins, well, understandably, represent contamination of the product. There are different reasons why these host cell proteins can be an issue. One example of these is that they can quite understandably provoke an immune response in their own right, since they are foreign material, either through the actual nature of the protein itself or possibly the glycosylation that is found on that protein. They can also possibly act to compromise the drug itself, so they could have an adverse effect on its efficacy and potency by modifying it in some way, such as protease-type enzymes. So quite understandably, HCPs need to be removed to the extent possible through the purification process, but also it's important that we monitor the levels of HCP to show that the purification process is robust and that nothing untoward is coming through into our product during manufacture. So at that point, we come to our first poll question, and for that I will hand back to Ernest.
Thank you very much, Richard. It's now time for our first polling question that we'll be running in today's session. Please select the answer that is relevant to you and then click submit. The first polling question reads, how do you plan on assessing HCPs in your development projects? By ELISA, by mass spectrometry, by ELISA and mass spectrometry. Richard, what kind of answers are you expecting here from the audience?
Well, this is going to be quite an interesting one, Ernest. The usual analytical procedure is ELISA. That's the one that is most heavily used. However, there is now very much a groundswell of opinion, and particularly from the regulatory authorities, that the use of mass spectrometry is very important to provide orthogonal and supportive data for the information that's generated by ELISA, particularly as the ELISA kits on the market will not necessarily detect all of the HCPs that are present in the sample that's being generated, because obviously each production process is fairly unique. So they tend to be taking the broad and most common HCPs rather than necessarily exactly what is related to the client sample. As such, a lot of HCPs can be missed by ELISA, so mass spectrometry provides supportive orthogonal information to detect a different subset of those HCPs. And by combining an ELISA analysis with a mass spectrometric analysis, you stand the greatest chance of covering the widest range of HCPs that are found in your sample.
Thank you very much for your added comments there, Richard. Let's reveal what the audience have answered.
Okay, well that's good to see. We have just over 40% saying by ELISA, 12.5% saying by mass spectrometry, but just under 50% say by ELISA and mass spectrometry. So that's very good to see that the idea of using mass spectrometry as an extra technique for HCPs is certainly being picked up. Okay, so if we just start to move forward into this now, just to tell you a little bit about how we can use mass spectrometry to approach the idea of quantitation of HCPs. First of all, in fairly simple terms, initially a digest is performed on the sample of interest using a specific protease or several different proteases in several different experiments, if appropriate. The products of digestion are separated out by online liquid chromatography mass spectrometry, and either MSMS or MS to the E analytical procedures are used to fragment the peptides as they elute off the system. MSMS, for those of you who aren't aware, is effectively a mass filtering system that is used to select different specific masses, fragment them and identify the fragment ions generated. MS to the E is, broadly speaking, an equivalent form which is used by Waters instruments such as the QTOF, whereby high and low energy fragmentation procedures alternate, and so rather than specifically focusing on particular masses, fragmentation takes place across everything that's eluting at a particular instant in time. So initially we take a digest mixture, we analyse it by mass spectrometry, and we are looking for various different marker peptides for HCPs out of this. In parallel, we would also look to be analysing the products of proteolytic digestion from isolated spots off of 2D gels. The reason for this is that the mass spectrometric analysis will allow you to identify what those components are off of the gel, but at the same time, if the HCPs aren't generating an immunological response, and that you will therefore not see a spot in the Western blot, but you can identify what those spots are nonetheless from a mass spectrometric compatible stained gel. So what this will allow you to do is generate more information about the HCPs that you find in your sample. So once we have this data, the fragment ion information that we generate from these procedures allows us to generate information on the sequence of the peptide. We can then search specific databases to look for the identity of the proteins. Subsequent to that, we can spike in to the sample a known amount of a known protein, and we can digest this mixture, separate by online LCMS again, and quantitate the HCPs based on the response of the HCP peptides or marker peptides and the protein that's been spiked in. This does provide quite a basic approach to quantitation in the sense that the different peptides will have different ionization properties. And so because they have these different structures and different ionization properties, that can compromise the accuracy of the data. To that end, we always recommend that we go to the next level as far as the quantitation of HCPs is, and that's what I'll describe now. So firstly, before we can go much further, we need to take a look at the fragmentation patterns of the kind of peptides that we generate in this analysis, and this will dictate the precise approach that we subsequently take. So here I've just shown an MSMS spectrum of a particular marker peptide identified in an HCP analysis, and you can clearly see from the parent signal at 957.5 there is a fragment ion at 715.38 Daltons. Now this, both signals are quite strong, there's good signal to noise present there, and these strong signals, these strong transitions, can be used to quantitate the amount of protein that's present in the sample. The stronger the transition we see, in other words, the greater the fragmentation, the more sensitive will our quantitation ultimately become. So how can we use this transition to help us? Well, this is the procedure here. First of all, we would identify the sequence of that peptide from the data generated, and we would subsequently produce a heavy cold-labeled analog of those peptides. Once these are synthesized, we quality control them to make sure that they're robust and that they'll produce a linear response in the mass spectrometer. Now, the reason for producing these heavy labeled analogs is that they will have exactly the same structural identity as the marker peptide of interest. As such, they will fragment in exactly the same way as the marker peptide of interest. The only difference will be that they will do it at a different mass. As such, we can use the mass spectrometer to identify these different masses of the same molecule effectively. The variation in ionization efficiency is therefore negated by the fact that these molecules are exactly the same. So, once we have our synthetic peptides, we will set up multiple reaction monitoring transitions, which is the means by which we can select for the transitions that we've identified in terms of the mass spectrometric data generated from the peptides. And these are developed for the native and the marker peptides, and for the heavy label peptides using either a triple quadrupole or a tandem mass spectrometer. The reason we use these instruments is that they have a large linear dynamic range, so we can quantitate accurately. We spike our samples with heavy label peptides. We then perform a digest. We separate out the components on our triple quad instrument, and we use the MRM, multiple reaction monitoring procedures that we've set up to identify and quantitate the peptides based on the peak areas that we are detecting in the mass spectrometer. And like I say, because we are dealing with structures in terms of the label peptide and the reference peptide that are identical, any differences that we would have had in ionization are ruled out. We can quantitate much more accurately. And using this technique, we can go to sub-ppm levels. Just to give you an example of the kind of data that we generate, the upper trace shows our LCMS data of the original digest mixture taken from a QTOF. And below, on our triple quad system or tandem quad system, you can see the data that is generated from filtering out all of the peptides that we're not looking for by using selected mass monitoring of our native and our heavy label peptides. The peak areas can then be determined, and based on the data that we generate and the known amount of material that we started with, we can calculate back the quantities of the individual peptides and therefore the individual proteins that are present in the sample. Now, the use of label peptides does extend beyond HCP analysis, and at BioPharmaSpec we've used these as well in PK studies of antibody-drug conjugates. Now, a discussion of that is really not what the webinar is about today, so I'm not going to be talking about that. However, we do need to talk about antibody-drug conjugates from another point of view. So just first of all, to give you a little bit of background on these. Antibody-drug conjugates, or ADCs, are produced by the chemical modification of antibodies such that specific drugs are incorporated at specific sites of the antibody chain through a chemical linker. Most commonly, these are cysteine or lysine because they're readily available as chemical entities for modification. The antibody then effectively is targeting molecule for those drugs to the site of action. However, we are relying on the chemistry for this, so any incomplete conjugations will result in products that are less potent, but they could even have their own adverse properties such as inherent immunogenicity or aggregation. So the efficiency of the chemistry needs to be monitored in order to make sure that the chemical procedures are optimized to produce the maximum amount of drug load on the antibody. And this idea of drug loading on an antibody is known as the drug-antibody ratio, or DAR. So we analyze molecules, or ADCs, here at BioPharmaSpec. If we're analyzing a lyse-modified form of an ADC, we would use the procedures listed here. We would analyze the molecule by LCMS in its native state as we receive it, also following a reduction to produce light and heavy chains, again following a DM glycosylation step to minimize the heterogeneity of the intact molecule, and subsequently combining a reduction and a DM glycosylation step to minimize the heterogeneity of the heavy chain alone. Just to give you an idea of the kind of data that we generate from this, this is an example using an antibody that was supplied by ADC. We did this work in collaboration with them. It represents a lyse-conjugated antibody that has been deglycosylated. The data that you see on the left represents the online LCMS separation or purification of that deglycosylated antibody. And you can see that the region that's boxed in is just looking like a very broad hump. However, the beauty of the QTOF system is that it's very highly sensitive and very high resolving, and as such, that data that appears to have very little information in it can be taken, passed through deconvolution algorithms, and produces a deconvoluted mass trace shown on the right. We can now see that that data is composed of a native monoclonal antibody at about 145 kilodaltons, and labelled 1, 2, 3, and 4 on top of that are different degrees of conjugation of the drug onto that antibody. And from the data that we've obtained with these conjugates, it gives a DAR of approximately 1.5. However, you can also see there, and these are marked by the green stars, another form of modification to the molecule, and this represents the presence of a hydrated linker. So therefore, we're looking at incomplete conjugation of the drug to this molecule. So the linker is bound, but the drug has not bound to the linker. Now, I said that both lysine and cysteine can be sites of conjugation for an antibody. Here is some data generated from cis-conjugated forms of an ADC. In this instance, we use a digest strategy to identify regions of the molecule, and we use the so-called IDES enzyme for this. This cleaves at the hinge region of IgGs in a similar way to papain. The fragments can be separated out, and here we show the online LCMS data generated from the heavy chain N-terminal segment of the ADC. And you can clearly see that we have no modified heavy chain, singly and doubly conjugated forms of the drug. And in each case, the linker is present but fully conjugated by the drug. So although the levels of binding of the linker are variable, the binding of the drug to the linker has been very successful. This work, as I should say, has been performed in collaboration with Glycera, both what I'm showing now and the previous slide. So this example here is a different antibody, handled in exactly the same way, so IDES digestion, reduction, and online LCMS. And here you can see we're not just seeing different forms of the heavy chain and drug, but we're also seeing different forms of bound linker. So here, this is implying that both the chemistry of linker conjugation and the chemistry of drug conjugation needs to be reviewed so that the yield is optimized. So you can see that this kind of analysis demonstrates that impurities are present in these samples to quite a large extent, and they can be minimized by going back and looking at the actual chemistry of the procedures that are being used. Now, whilst that's effectively a deliberate form of chemical modification of the molecule, there are a lot of other molecules. There are a lot of other types of modification and impurity that are the result of unexpected or non-deliberate forms of modification of the molecule. A lot of these are listed out in the process-related impurities section. I'm just going to use a few examples here. The first one I'm going to talk about is scrambled disulfide bridges. These represent, for those of you who don't know too much about protein chemistry, disulfide bridges are simply the thiol crosslinks formed between two cysteine residues. Because two are involved, it's called a disulfide bridge. The cross-linking needs to be correct in order to stabilize the molecule. If that cross-linking is incorrect, a misfolded protein will result. This can be inactive, possibly in itself immunogenic, or even susceptible to aggregation. So it's important that disulfide bridges and protein folding is correct. Incorrect cross-linking is known as scrambling. And these are regarded as product-related impurities. Now, we use a procedure of peptide mapping to assess for scrambled disulfides. Peptide mapping is a very powerful technique for analysing not just the peptides and identities of those peptides out of a molecule, but because it uses mass measurement, it can identify many other modifications associated with those molecules, and therefore is an ideal tool for impurity analysis. We perform this work by digesting under non-reducing conditions using a protease appropriate for the molecule under investigation. Again, separating out by online LCMS procedures and analysing the data using a QTOF instrument. This is beneficial because we can generate fragmentation information by the MS to the E mechanism. As such, what we don't need to do is effectively repeat an analysis with a before and after reduction step, which would be needed if we were looking for disulfide bridges and their subsequent loss following reduction, with the production of two other peptides representing either halves of those bridges. So MS to the E provides us a much more rapid way of assessing bridge structure. Here I just show an example of some data that we generated from the peptide mapping analysis of a particular molecule. The upper signal shows a triply charged peptide. Here we have three peptides actually linked through two disulfide bridges. The bottom spectrum shows a 7-charged molecule. This is actually 4 peptides that are linked by 3 bridges. And those represent the correct structures that we'd expect to find in the molecule. However, we did find another signal in there, which was that shown here. So this again is a triply charged molecule or signal at 603 Daltons. We suspected this could be indicative of scrambled disulfide bridges. However, the way to confirm that was to look at the high energy MS to the E data, and when we looked at that, we saw a whole series of fragment ions as shown in the trace below, indicative of cleavage down the peptide backbone of the molecule. The majority of ions were generated from the C termini of the peptide, so-called Y ions. One B ion, representing the N terminus of the peptide, can be seen. And these peptides are consistent with the sequences shown and the peptides present, and therefore indicate that this was indeed a scrambled disulfide bridge. So the peptide mapping has been very significant for this kind of analysis, and of course other bridge possibilities can be assessed in the digest. Now we come to the next poll question, and for that I'll hand back to Ernest.
Thank you very much, Richard. We'll now be running our second polling question in today's session. The polling question reads: have process or product-related impurities impacted upon your product development? And the selection of answers are yes, yes but not significantly, no, or yet to be studied. Richard, what are you expecting here from the audience?
I would think that probably the second answer would be the one that would be most expected. I would think that people would have a degree of impaction from the impurities in the sample. But when you know it's there, you can work around that and then produce a molecule that does have the quality you need. So I would say that assessing the molecule is very, very important in order to find a way to deal with those impurities and produce the quality. So I would think that the second answer would be the most common one.
Great, thank you very much. Let's see if this is correct. Oh, very interesting, Richard. What are your thoughts on that?
Let's just bring that up. Yeah, that's kind of what I would expect, yes. Maybe we've got just under 50% saying yes, but not significantly. About 35% saying yes. I'm surprised that there's so much in the yes camp, but it could be that that there have been ways found to deal with the results of impurities. I'm not surprised that there's 0% for no. And obviously, those who are still yet to study it, well, I guess we'll see. Okay, so moving on, I'm just going to talk about 2 more examples of chemical modifications to proteins. The first one is oxidation. Again, this is a very common form of modification. It could be produced through the production process itself, or maybe is reflective of a lack of stability in the molecule as well. Very often it occurs at the side chain of methionines. Because it's a chemical modification, it results in a shift in mass. The attachment of a single oxygen on the side chain of methionine produces methionine sulfoxide with a mass increase of 16 Daltons. Should we get a double substitution, we produce so-called methionine sulfone with a mass increase of 32 Daltons. Now, one of the things that you notice about oxidized methionine is that it can actually fragment down its side chain. And this is very useful because we can use the loss of 64 as a fragment ion to assess the data and look for oxidized species. And I'll show an example of that next. The chemical modification of methionine doesn't just affect the mass, but it also affects the chromatographic retention properties of the peptide. So what that means is that by LC-MS procedures, we can separate out native and oxidized forms of the molecule. Now, here I show an example of an online LCMS chromatogram, and the bottom tray shows the total ion chromatogram. In other words, that's the mass spectrometric data indicating the presence of all peptides. Above it, we have the extracted ion chromatogram, where we specifically search the data for the mass of the native peptide alone. And above that, we have the extracted ion chromatogram for the mass consistent with the oxidized peptide. Now, we can take those peak areas and we can integrate them, and we can therefore use that as a measure of the percentage of oxidation in the molecule. However, any differences in ionization between the oxidized and native forms is not taken into account in this analysis. However, for purposes of comparability between samples, it's not an issue, since that difference will cancel out between the different samples. What we do need to know, of course, is to make sure that we're analysing, that we're looking at the right thing, because at the moment we simply have a peak and a retention time. However, once again, we can go to the MS to the E data, and we can look at that for confirmatory fragment ions to tell us that we're looking at the peptides of interest here, and that therefore the quantitation is appropriate. The lower of the two spectra you see here represents the native peptide in the previous spectrum, and we can see fragment ions through that spectrum consistent with the C-terminal amino acids of this, the so-called Y ions, produced by cleavage again down the amide backbone. All ions consistent with the masses of each amino acid, as we would expect. However, if you look at the upper of the two traces, this is consistent with the oxidized species. We can see that there is a different series of fragment ions. All of those are offset once we have a residue containing the methionine species. So they're all 16 Daltons higher than the spectrum below, consistent with the presence of an oxidized methionine. And we see the signal from 322.2 Daltons to 258.2 Daltons, representing the loss of the 64 from the side chain of the methionine itself. So this is confirmatory evidence that the signals we were looking at were indeed reflective of the peptide and its oxidized form. Now, we can do a similar thing with another very common modification, and that's deamidation. This can occur at the side chain of the amino acids glutamine or asparagine, and it can be identified by a mass difference in the mass spectrometer because we are chemically modifying the molecule. The modification is relatively minor. It's a single Dalton, simply because we've converted the NH2 function of the amide to an OH function in the acid, as shown here. I show asparagine, but exactly the same thing happens in the case of glutamine. Now, it is possible to analyse deamidated forms using other procedures rather than mass spectrometry, and the classic one would be imaging capillary isoelectric focusing. Because we've modified the side chain from an amide to an acid, we effectively change the PI of the molecule. So we can look for changes in PI as a measure of a change in the structure. And that's what we show here. This is an example of an antibody. It's run between PI marks of 4.2 and 9.5. The trace on the left, the electropherogram on the left, shows the full scale. The one on the right shows the zoomed-in region of the antibody. The antibody main component has a PI of 9.1. There's a small signal at 9.2, which is consistent with the attachment of an extra lysine on the C-terminus of the heavy chain, because there is lysine variability in antibodies, of course. And the signals that are slightly more acidic PIs of 9 and 8.9 will be consistent with deamidation of the molecule. Now, this is all very useful as a rough guide, but it's not telling you anything about specifically what is going on within the sample or where this is happening, because this is just the summation of a net change of PI on the molecule. But we can carry on with this analysis by going back to our old friend, the peptide map, and we can analyse these molecules in the same way as we would for oxidized forms. So we can take our molecule, we carry out a specific digest, separate out the components, and once again we have the TIC showing the total peptides observed at the bottom, and then the extracted ion chromatograms for the native and the deamidated peptides shown above. Again, we can take the integrated peak areas and work out a percentage of deamidation based on that, but we suffer with the same issues, of course, as we would for oxidation, in the sense that the differences in ionization between deamidated and native forms are not taken into account with this procedure. But once again, for comparability purposes, that is nulled out across different samples. So we still need to prove that we've got the the peptides of interest, and once again we go to the high energy data for this. The peptide trace at the bottom shows the Y ions exactly as we would expect from this peptide. Trace above, although it appears weak, still has good signal to noise when you look at those peptides. Other peptides that you see present are generated, other signals that you see present are generated from co-eluting peptides, because this is an MS to the E trace rather than an MSMS trace. Nonetheless, we can see a series of Y ions that are consistent with the first three amino acids. However, once we get to the point of deamidation and the conversion of the glutamine to glutamic acid, this causes a mass shift of 1 Dalton, and the subsequent fragment ions are shifted accordingly. We therefore have confirmation that this is the peptide and the deamidated forms, and so the quantitation is being carried out appropriately. I just want to touch briefly on carbohydrate analysis. So we've talked a lot about proteins, but obviously that's not the sole cause of contaminants for samples. Carbohydrates can be contaminants as well, particularly things like during gel filtration processes, breakdown can give problems there. A very simple way for analysing or assessing this is to use the monosaccharide analysis by GCMS, and this is something that we routinely do at BioPharmaSpec. A lot of information can be generated that tells you both about the molecular or the glycans that are present on your molecule, as well as maybe whether other contaminants are present or not, based on the signals you see and the intensity of those signals, because this is a technique that we can quantitate using known standards and the responses of those known standards by GC. Just to give you an example of that data, if you look at these two GCMS traces here, the lower profile at the bottom shows the expected data, where we see high levels of mannose. Fucose is also present. Some glucose is present, as would be expected, to be honest with you, because glucose is a very common contaminant. It can come in through reagents, glassware, etc., etc. So very often there will be a level of glucose in the sample. The erythritol that you see there is the internal standard for quantitative purposes. If you look at the observed profile, you can see that it's very different. The mannose levels are significantly decreased relative to glucose, which is very considerably increased over the expected profile. This was consistent with breakdown of Cephalodex products during the purification process and clearly demonstrated that purification needed to be rethought. This is the kind of data that I have seen on several occasions. I've also seen it in instances where yeast cells have been used. Purification has not been as expected in terms of its efficacy, and very, very high levels of mannose have been observed. And again, that means going back to the drawing board, reassessing purification, and then moving forward again. So finally, I'd just like to talk a little bit about aggregation. We've talked a lot about the actual molecule itself and problems that may be associated with that. One of the larger problems that can be associated with molecules is aggregation itself. Now, this is a very important topic for all the regulatory agencies. I've just shown a little bit of the text here for both the FDA and EMA biosimilarity guidelines. And you can see for the FDA, they mention aggregation, as you would expect. And they say the identification and determination of the relative levels of these protein variants should be included in the comparative analytical characterization studies. They specifically state orthogonal methods should be used to assess aggregation. This is effectively mirrored by the EMA. They say qualitative and quantitative combination of analytical procedures should be used using orthogonal and state-of-the-art methods, and they specifically state aggregation in their list of comparators at that point. So we use two methods for this analysis. The first one is SEP-MALS, which is size exclusion chromatography with multi-angle laser light scattering. This uses a column-based separation technique with detection using UV, refractive index, and MALS detectors. The reason for the use of UV and RI is that larger particulates can scatter rather than absorb UV light, so we have to make sure that we're detecting all forms of aggregates to the extent possible. The separation and use of molar detectors allows us to determine the molar mass of the components as they elute, and we can quantitate the monomers and aggregates that we see. And you can see in the data presented in the figure there that we have a monomeric species of about 150 kilodalton and a small amount of a dimeric species of about 300 kilodaltons. Now, this is a column-based technique, so it does suffer from issues associated with column analytics. This means that turbidity and shear forces can come into play. That can cause a disruption of aggregates, and therefore a lower level of aggregation can be observed. As an orthogonal procedure to this, we use SVAUC, which is sedimentation velocity analytical ultracentrifugation. In this instance, we centrifuge the samples at very high speed, and in conjunction with centrifugation, we use UV detection of the centrifuge tubes over time to measure the sedimentation of the components that are in the sample. Again, this allows quantitation and mass determination by taking the data, passing it through algorithms, and translating that into the different levels of the different components and their sedimentation coefficients. It does have the advantage that it's a column-free technique, and this is highly favoured by agencies such as the FDA. What it does mean, though, is that because you're using high centrifugal forces, you're in effect concentrating the sample. So this can lead to a higher value for aggregation as you effectively force those molecules together. So the use of SpecMols and SVAUC will give you an upper and lower limit, but the data that you generate will be dependent on the nature of the technique that you're using to generate that data. So finally, just to summarize this, impurities have the capacity to adversely impact product quality through a whole variety of different procedures, most commonly immunogenicity, aggregation, or potentially a reduction in efficacy and potency of the molecule itself. Broadly speaking, impurities can be placed into two distinct groups. These are the product-related impurities, which are adverse alterations to the structure itself, and process-related impurities, which are those that are present within the product as a result of the processes employed during the manufacture. Regulatory authorities require a detailed understanding of product and its impurities. Quite understandably, they would expect you to know your product, they'd expect you to know what's in your product, they'd expect you to control what's in your product and minimize the impurities that are present. There is an expectation that state-of-the-art analytical techniques are used for this investigation. And finally, successful product development and production relies on monitoring the product and the associated impurities so that decisions are taken based on the data, and all of the procedures associated with manufacturing the molecule of interest can therefore be brought back to a data-driven decision and monitored in appropriate ways with appropriate state-of-the-art analytics. So finally, this takes us to our last poll question for the day, and I'll hand back to Ernest for that.
Thank you very much, Richard. Indeed, it is our last poll question for the session. Please select the answer that is relevant to you and then click Submit. The polling question reads, what has been your major challenge in dealing with impurities? And the selection of answers are HCPs, other contaminants associated with sample purification, other protein modifications, or glycosylation modification. Richard, what are you expecting from the audience from these answers?
Well, this is going to be an interesting one, Ernest. This will be very much dependent on the products that are being developed. Everything will effectively have its own level of impurities, particularly as at some point you have to define what your product is, and as such, anything that falls out of that product definition is in effect an impurity. So it will depend on the nature of the molecule being produced and the stage of production that we're at. So it's going to be very hard to say. I'm just really quite curious as to what people say to this one.
Thank you very much. Let's see what the audience have chosen. more have gone for HCPs. Is that what you were referring to, Richard?
It doesn't surprise me. Yes, so 50% have gone for HCPs. Any biological molecule is going to have HCPs associated with it, but they have to be monitored, they have to be minimized. They are a challenge, they're a big challenge, because of their diverse nature. You're dealing with a huge range of different types of molecule here, and they have to be accounted for as far as possible using, as I say, ELISA in conjunction with mass spectrometry is the best coupling of procedures to give you the widest spread. But it is a significant undertaking. You can have a lot of HCPs in there. That's a lot of impurities that you need to control and manage. Okay, so that brings us to the end of the webinar for today. Finally, I'd just like to leave you with some information about us. I'm at the top there. That's my contact details. So if you need to get in touch with me or you have any other questions, then please feel free to email me. We do have various business development partners around the globe, so their details are shown on the slide as well. Please feel free to contact your local agent who can put you in touch with one of us, one of the scientists on site, if they cannot answer your question directly. Thank you very much for your attention, and with that I will take any questions.
Thank you very much, Richard. We have received a good amount of questions from the audience. I'll start off by reading the first one. How can you explain how glycan profiling can be used to identify and characterize product-related glycosylation impurities?
Okay, yes, thank you for that question. We didn't really get a chance to touch on this just because of time. But of course, the glycosylation profile of your molecule is going to be very important for its own modes of action. That can change depending on the environment that the molecule is made in, the stresses that the cells find themselves in, pH, dissolved oxygen, all sorts of things like that. In simple terms, something like analysis of fluorescent derivatives such as 2-aminobenzamide in conjunction with online mass spec can generate a profile of the sample. That allows you to quantitate the different forms and identify the masses of the different components that are present. Should anything change in the profile, that can be readily detected in an analysis like this. But also, there are other mass spectrometric techniques that are available, and they're certainly employed very frequently by us here at BioPharmaSpec to characterize what those changes are. So should something untoward appear in a profile, other techniques can be brought into play to determine accurately how, or rather what the mass of these different components are, these unusual components are, what the linkages of these different components are, and therefore identify what these contaminants in glycosylation turn out to be. So the profiling can start with something that's relatively simple, but then can move down to more complex techniques to identify exactly what these impurities turn out to be.
A very detailed answer there, Richard. Thank you. The next question that we have here from the audience reads, is there a benefit to using a CRO for work like this when I have a mass spec in-house and could develop methodology myself?
Yes, I would say there is certainly benefit in using a CRO. Whilst it's true that methods can be developed in-house, very often you will find that a CRO may well have prior experience of that kind of work. They will most likely have techniques in place. We certainly have techniques in place for analysing impurities, and hopefully that has been shown today. Also, there is the idea of orthogonality as well. Other techniques can be brought to bear to provide supportive data for the data that has been generated and thus give greater veracity to the information that is obtained. CROs can also act as overspills potentially as well, and they certainly have an awful lot of experience in handling impurities. So very often it can be easier simply to outsource to a CRO than to spend time developing a method in-house. If it is needed to have a method brought in-house, then again, a CRO can develop that method and then transfer it back in through a tech transfer arrangement. And again, that's certainly something that we at BioPharmaSpec can do.
Great, thank you very much. Thank you for all your questions so far. If you would like to submit a question, please use the Q&A box and we'll try and get through as many of your questions as we can. The next question reads, is there a best time to start assessing impurities in a product?
Yeah, that's a good question. The best time to start assessing impurities in product will really be right from the outset of the work, I would say. But the nature of that assessment will change over time. For example, there is a requirement nowadays, certainly in the area of biosimilars, that the full amino acid sequence of the molecule being generated needs to be confirmed at the amino acid level. So we're not talking about just the DNA sequence, but actually at the amino acid level. Now, as such, that may mean that certain sequence variants need to be looked for based on the inoculation media that is used in the various different amino acid derivatives that may be available. So sequence variant analysis may be something that is needed at the early stage because that effectively is an impurity. Glycosylation profiling we've already talked about, but that is another kind of classic example where it is very sensitive to system change, and you need to show that initially you're producing something from that system that is correct. Anything outside of that, again, could be an impurity. Now, that doesn't mean that you spend all of your time analysing impurities right from the outset, because that would be rather pointless. But you will find that as you go through product development, the need for these kind of impurities will change. The types of analyses that you perform will change in line with the production process. So I would say really impurity profiling starts from the very beginning of the work, but is focused on exactly the requirements at that time and will develop along with the development of the product itself.
Great, thank you very much. We've received an interesting question here. It reads, are procedures for impurity analysis very generic, or are they product specific?
They are. Well, it's kind of a mixture of both, really. I know that's a bit of a kind of a cop-out answer, but really, what you're looking at is a series of broadly similar techniques that have to be applied in certain ways depending on the nature of the molecule you're looking at. For example, something like the peptide mapping that we've talked about quite extensively today, that can be used for analysing any glycoprotein. But the particular way that you produce the peptides will depend on the nature of the molecule that you are examining, because the amino acid sequences will all be different. So there is a large degree of commonality in the procedures, but very often, or depending on what you're precisely examining, you may well need to have a degree of specificity or tailoring to that particular molecule so that you can get the maximal data out of it.
Thank you very much for your answer there, Richard. We now have another question. How does the analysis of product and process-related impurities develop with product development?
Okay, yes, thank you for that question. That kind of sort of feeds back to something we touched on earlier. There will be a development in terms of the kind of techniques that are used during the assessment of impurities. At the early stage, there will be a very limited set of tests. However, as you move forward and as your production process develops, there will be a much greater onus on assessing the impurities, such as HCPs, such as assessments of things like the glycosylation, or rather the carbohydrate contaminants that I talked about earlier, to show that you're getting no problems with breakdown of any of the consumables or reagents that you're using during the process. You also need to show that when you're producing a drug substance and your drug product, by the time you've got to that stage, the production process is such that you're not introducing any contaminants such as a lot of disulfide bridge scrambling or a lot of oxidation or deamidation to the molecule. So you would start off quite small, but by the time you've got to a full-scale characterization of your product, you should be looking at a full-scale characterization of the impurities associated with that as well, and showing that in terms of batch-to-batch production, those impurities are not changing, and therefore you have control of your manufacturing process, which is something that the regulatory authorities will need.
Thank you very much for a detailed answer there, Richard. And thank you all for your questions, but unfortunately, we have run out of time. If you have submitted a question, BioPharmaSpec will respond to you within due time. So that just leaves me to thank Richard for what was a great presentation, and thanks to BioPharmaSpec for sponsoring this session. To the attendees, you will receive an email tomorrow telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-reviewwebinars.com. Please continue to ask questions, vote in polls, and interact with the widgets as the on-demand version remains interactive. We look forward to sharing further webinars with you, so please keep an eye out on our website and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again for tuning in, and I all hope you have a great day. Thank you.